TN2 is the translational neuroscience network that we have in Amsterdam Neuroscience, aimed at bridging the two different fields, namely very clinical and very fundamental work. So this seminar is recorded and is, as you can see, in Zoom. And as it is a webinar, all of you cannot see each other. And we can also not see your video, but what you can do is participate in the Q&A. So you can send us your questions using the button that you can see here. Don't use the chat option for this because we really want to use the Q&A for the questions. Now, what we'll do is while the speakers are busy, we will monitor the chat, the Q&A, and then we will read out the questions to them. What you can also do is if you want to change the graphic interface, you can slide this little line back and forth to make the speaker a bit bigger or the slides a bit bigger. So you can customize things a little bit. All right, so what I would like to do now is hand over to, to introduce a little bit more TN2. So as I said before, it is a bridging between fundamental and clinical neuroscience. We have annual conferences, of course, before Corona. So unfortunately, we didn't have an in-person conference in a while now, but we will do again once things settle down. But in the meantime, we have these lunch seminars. We also did this last year, and now we have a series of 9 webinars every month until June, and every month now we have a different research pillar that is the topic. You'll see in a second what the pillars are. So please feel free to join the other meetings as well. You don't have to just join your own research pillar, and spread the news if you want other people to join seminars as well. So now I will hand over to my co-chair, Odile van Heuvel.
Yeah, thank you, Menno. Welcome to this CIA-oriented TN2 seminar. As you can see, we are this yellow pillar, the CIA, Compulsivity, Impulsivity, and Attention. So this is one of the 2 psychiatry-oriented programs within Amsterdam Neuroscience. So next slide, please. No. So half a minute, what is CIA? I think it's a good example on how to bridge fundamental research to clinical practice. And our fundamental goal is to use information from basic neuroscience to, in the end, inform us about what is the disease and how can we target the system in a better way to get better treatments. And the 2 speakers of today are exactly those kind of people that can bridge between preclinical research and clinical practice. So let's jump to them because they are the stars of today.
Okay, so now it is my pleasure to introduce you to Ingo Willuhn. Ingo is a behavioral neuroscientist with a special interest in functional neuroanatomy and especially the behavioral functions of the basal ganglia. And within the basal ganglia, especially the striatum, looking at different neurotransmitter systems. He's especially focused on rodent models, but also aims to translate his findings to a human setting. He's originally from Düsseldorf, but then did his PhD in Chicago, a postdoc in Seattle, and now came here to Amsterdam UMC and at the Netherlands Institute for Neuroscience, the NIN, where he is a group leader in neuromodulation and behavior, and is also heavily involved in the Amsterdam Brain and Cognition Center. And today he will discuss with you his latest work on brain mechanisms underlying OCD using deep brain stimulation in a rodent model.
Shall I go ahead?
Go ahead. Yep.
Share my slides and also my pointer. Well, thank you very much for the kind introduction. Yeah, so as pointed out, I'm leading a preclinical neuroscience lab, but we have very strong ties to the psychiatry department of the AMC, which is one of the departments that's world leading in OCD research and specifically also in the treatment of OCD patients with DBS. Can you see my slides?
No, we can't.
Oh, that's
I think you have to reshare them.
Okay, I'll share again. Here we go. And this should be better now.
Yeah, now we can see them.
Perfect. Sorry for that. So yeah, yeah, we're connected with the clinicians that treat OCD with DBS, and even though like it's been used very successfully, like here in Amsterdam, like we do know fairly little about what actually happens in the brain when DBS is switched on. So I'm going to tell you about our quest for like trying to find out more about this topic. And I'm going to mostly like show you data that was collected by my very talented PhD student, Martijn van den Boom. Like he really moved mountains for this project. So Martijn is going to talk a bit more about OCD itself, I think, but in brief, like we are mainly interested in the behavioral aspects, the compulsive behavior that is performed despite negative consequences by these patients. These are repetitive stereotype behaviors that are often perceived as a result of obsessions that say, for example, contamination fear that leads to hand washing. And these obsessions, together with the anxious phenotype of these patients, lead to the fact that these patients really can't stop this compulsive behavior, even though they would like to. It comes with a few other features frequently, which is, for example, decreased cognitive flexibility and altered habit formation. On the brain end, we have some good evidence that points to like a problematic communication between the cortex and the striatum. The treatment that is used as a first line is usually like a SSRI medication and cognitive behavioral therapy. But importantly, there is a number of patients, substantial number really, that does not respond to these treatments. And for those very severely affected patients that are very desperate, often suicidal even, like Damian Denise and his group here in Amsterdam has for over 10 years now utilized deep brain stimulation to help them. And here's like the head of one of those DBS patients during surgery where the DBS electrodes implanted very deep in the brain, as you can see here. And the tip of this electrode is usually targeted at the border between the nucleus accumbens, which is a striatal nucleus that is involved in motivation, and the V alic, the ventral anterior limb of the internal capsule, which is a massive fiber bundle that has fibers from and to the prefrontal cortex and has involvement like in planning and decision making. Now, this is a different view, like a 3D model of this electrode implantation. So in green here, you see the electrode being implanted really at the center of the basic ganglia, going through its core. The IC, the internal capsule, is here in brown, and then this dark brown here is the nucleus accumbens. So over the years, it has become clear that even though the initial target was the accumbens, that the most effective targets for treating OCD are actually further up near the internal capsule. So as I said, like this treatment works pretty well. You have to remember these patients cannot be helped any other way. So achieving up to 60% of response is a good success. But of course, it's not all of them. And also like the tweaking the parameters of the simulation can often take months. So it takes quite some time. And the way the optimal parameters are found is usually by a trial and error process. And so like the standard parameters are switched on, and then the clinicians look, is there an effect? If there's no clinical effect, then they increase the voltage of the stimulation. If there's still no clinical effect after doing that incrementally, the pulse width is changed. And then if no effect, then like even different contact points, like slightly different targets are used. So like the question for us is here, like how does it work in the brain really? And is there any way that we can help or assist to make this better? And that's where my group comes in. Like we use animal models to look at compulsive behavior. One of these animal models is the SUPAP3 knockout. SUPAP3 is a protein that's expressed in postsynaptic densities of glutamatergic excitatory synapses. So that's what you can see here. And it's primarily expressed in cortex and in the striatum. So exactly those regions that are affected in OCD. Taking this protein out, deleting it genetically, leads to some dysfunction in the communication between cortex and striatum, which is also a hallmark of OCD patients. On the behavioral end, these mice, they exhibit quite a few features that are similar to the patients. They have this very excessive grooming that leads to even loss of their fur if left untreated. So that's their compulsive behavior that they're performing. They have decreased cognitive flexibility, greater anxiety, and also altered habit formation. And just as in the patients, SSRI can help to normalize these deficits. Now, in a previous publication, we have implanted DBS electrodes the exact same place as in the patients in the internal capsule. This is what's portrayed here. And these are the standard parameters used in the clinics. What we found is, first of all, very trivial, that stimulating the brain with deep brain stimulation does affect and activate cells, which is shown here with CFOS around the electrode tip. And then I'm showing you a video of an animal that actually is grooming quite a bit. And the DBS stimulation is switched on in a few seconds. And you can see how this mouse goes from grooming to snapping out of this type of behavior and doing other things like exploring the box more. And this is just showing you that this, expressed in numbers, we can successfully suppress this compulsive grooming with DBS. So we know it works in general, but how does it look like specifically, and can we learn more than this? So what we did is we did a very systematic approach, which of course you cannot do in humans for ethical reasons, where we varied the current of the stimulation, we varied the pulse width, and we varied the frequency in different settings or in different conditions. And what you find is that the grooming response to the stimulation is a very nice dose response. So you see how applying more and more current leads to less and less grooming. And the red line here is the onset of DBS, and the other red line here is the offset. So you can really see this dip in between while stimulation is on. There's a fairly similar a picture when you change pulse width and you increase the pulse width, and also with frequency, but there are some small differences. So first of all, if you go below the standard stimulation that is used in the clinics, you don't see an effect on grooming. And then in yellow here, you see that the standard parameters, which are all the same between these three here, definitely leads to a decrease in this compulsive grooming, but only for the current increases and for the pulse width increases, you see an additional decrease that goes even beyond what you already got the standard stimulation, which is not the case with frequency. Now, how specific is this? So if you look at general motor activity, you see that DBS does not affect the overall ambulation of the mice. So it's not just that the mice just run around more and therefore they groom less. And if you do this to control mice that don't have the protein deletion, you don't see an effect on their grooming at all, which is their grooming is of course lower to begin with. Now to summarize these findings, we express the stimulation as a function of the charge that actually is applied to the tissue. So with all these three different parameters being changed, if you increase it, of course you increase the charge applied. And what I just showed you already is essentially that with current and with pulse width, if you increase the intensity even more, you also get more of an effect on the grooming. But that's not the case with the frequency. So which is an important point, because it means that just simply increasing the charge that you apply to the tissue does not necessarily result in a positive outcome. So it has to be in a certain way that you have to do it. Now, so what about the brain? So we know on the behavioral level, just like in patients, this DBS works, but what happens in the brain? So here it becomes a bit more technical. So what you see here is a brain of a Taiwan GCAM6F mouse, meaning this mouse is genetically manipulated, so it expresses GCAM in their neurons. And GCAM is essentially just a fluorescence marker for calcium. So whenever calcium binds, when calcium is around to this GCAM molecule, it makes this GCAM molecule make light. So it's green light that you can see. So you're really just making neuronal activity visible as green light. And what you then do is you can make the skull of these animals see-through, and you can put a microscope on top of it. So you can actually see the entire dorsal cortex of these mice, and you can see neuronal activity across this entire mantle of cortex. And then what we did, of course, is we stimulated with the DBS in the internal capsule to see what happens to this dorsal cortex activity. And that's pretty much what's shown here. So here is the top view. This is what the microscope sees essentially. On the right side here, I'm going to show you a video in a second. And on the left side, you see this dorsal cortex partitioned into different functional units. And whatever is red is an increase in activity of the neurons, and blue is a decrease. So you're going to see here DBS going to apply it now. And you see that there's the blue in the overall cortex is actually increasing. So there's a decrease in activity. And then it returns more to normal after the stimulation is switched off. And if you look at the different parameters that we use, you see that with increasing current, this dip in activity actually increases. And this different activity is actually more prominent in the frontal regions and in the more frontal regions of the brain, and not so much in the visual and retrospinal cortices. Similar with pulse width, even more extreme. You have a more frontal effect, and less so with frequency again. So this is consistent with what we found before on the behavioral level. Now, with this approach, we weren't able to look at individual cells, and we wanted to really look at individual neurons. So for that, we used a different approach. It also utilizes this GCAM protein, but in this situation, it's expressed in specific brain regions by a virus injection. And then the animals are implanted with with lenses and essentially like a mini microscope that then can catch the neuronal activity, the fluorescence, while the animals are moving around. So this is what you see here. So here's a mouse, and at the same time, we can see the cell activity in regions of interest. And at the end, you have like about 100 neurons per brain region, and you can see what each individual neuron is doing during DBS. Now, yeah, here's the general setup. So again, like this is a coronal mouse brain section. In yellow, here's the internal capsule, and this is where our stimulation is. And this is then when Martijn, the PhD student working on this, showed us true colors. Like he, instead of one brain region, he did 6 brain regions that are all relevant for OCD. So this gave a great wealth of data just to show you how wonderful of a data set this is. I'm just showing you example videos for all of these different regions, and you can see the neurons light up while animals are behaving. Now, as a first step, for each of these brain regions, I just plotted here if you just average all the neurons, and then you have the different intensities for current, in this case, and red is the strongest stimulation, blue is no stimulation, you see that there's a peak at the beginning when stimulation is switched on, and then there's not a whole lot of differences between the different stimulation intensities, except for maybe the stratum, where there's something that is more sustained. So that is sort of what you would see, I guess, also with let's say fMRI. Wherever you look at a population signal, you would see this. But we now had the chance to look at individual neurons. So that's what we did. We classified neurons as neurons that actually increase their activity in response to the DBS stimulation, starting here, and neurons that are actually decreasing. And you see with more and more stimulation intensity, you also get a higher response here. So this activity would be masked if you only look at the average, of course. And then we went a bit further because we said, okay, there is this initial peak after stimulation onset, but we're not really that interested in it because we think the important part are the sustained recruited neurons. And when I say recruited here, I mean either deflected and having more activity or deflected down and having less activity. So they're reacting to the DBS stimulation. And this is what we think might be more important for permanent changes in grooming activity. And also in patients, DBS stimulation is switched on for very long periods of time. And this is a complicated graph I'm just going to summarize for you, which essentially is saying the more stimulation you apply, both the more neurons you recruit, but also the more the average activity of these individual neurons is affected. That's an interesting point for later. Now, to compare different brain regions with this, I just made these plots, or Martijn made these plots, I should say, where again, we're talking about recruitment of neurons, and that includes these 4 different categories. And essentially, in here, you have the transient increase, and what you see is that with more and more stimulation intensity, more neurons are recruited, is what we had sort of expected here. And the other important point, which I think is really crucial and something that is worth discussing later, is that you see there's more and more recruitment with more intensity, but the yellow and the blue bits are about in the same ratio. That means that for each neuron, so to say, DBS recruits that is excited by DBS, it recruits also a neuron that is actually inhibited by DBS. And this is mainly pyramidal cells in this case. So I kind of refer to this as a stolen word from EEG research, the excitation inhibition balance. So the brain region actually maintains about the same balance between these two, even though there's more and more neurons involved. And you can also see this in all the other regions. There is this dose response, and the blue and the yellow bits are more or less the same. The same is true also if you change pulse width. Now, to make this a bit more digestible, we plotted this also just for each brain region by itself, and we only looked at the sustained neurons now. And what you see is that, even though all the brain regions show a similar effect, there's some that are sticking out. And it seems like that the OFC specifically has more of a sustained increase in other regions. And the dorsal striatum shows definitely an increase, but doesn't show a decrease in cells. So here's like where this like balance might not be the same. And the same is true if you look at pulse width. So like the nice thing is we can look at these different parameters to actually do like a reality check. Do we see this, you know, with different types of effective stimulations, pulse width changes, or the current changes? Yeah, this graph is, well, I like to always like make people imagine you're a prefrontal cortical neuron, and imagine DBS is switched on and you're around. So, and you decide to respond with more activity to DBS. How about the next time DBS is switched on? Like, are you actually doing the same thing? And this graph just shows you, like, once you're on the team of being a neuron that is excited by DBS, you keep doing the same thing. You're not going to switch over and be sort of inhibited or suppressed in your activity the next time. Again, the same thing happens with pulse width. So that's something like that's important in terms of consistency of reactions of these neurons. But how about their anatomical like relationship? And what you see here is like just 5 neurons marked that are reacting to DBS, and these are in closer proximity to one another than others. And you see that there's more what you could call local functional connectivity between them. So they're doing more of the same because they're closer to one another. And that's also true if you look at these 3 neurons like together. And then we compared this like across the brain regions by like comparing to a shuffled like response. And what you see is essentially in all these graphs, the exact same thing, is that the closer the neurons are together, so these are neurons that are closer together, the more correlated their response, and the further they're apart, the less they are doing the same thing. But the important point is really here that DBS doesn't change anything about this. So like this local functional connectivity is completely preserved. Like no matter how hard you stimulate, no matter which brain region, there's no change in this. And as one of the last points I want to show you is that also taking advantage of the view that we actually can see where those neurons are. So on top, in the top row here, you see neurons that are increased in response to DBS at different stimulation intensities. And on the bottom row, it's like the neurons that respond with the suppression. And like one question for us was just simply like, well, are these neurons sort of like bundled together? Are they all bunched up in the same place? And the answer is these like responding neurons, they're really scattered throughout the brain region. So that's an important point, I think, also when considering like how this actually works. So like with this arrow here now indicate the same cell in different conditions, and this same cell is activated every time, and this same cell down here is decreased every time DBS is switched on. But there are also cells just come on randomly, it seems, in different conditions, but not the other. So as a last thing, what we wanted to do is to see like what is the consistency of response. So if you're a prefrontal cortical neuron, we know already you are either an excited or an inhibited neuron, but how often do you actually do this? How consistent is this response? So we made these Venn diagrams, and that just shows the number of neurons that are excited by 200 microamp stimulation, 300 microamp stimulation, and so on. And then the overlap is here, the neurons that are actually the same between the two. So what you can already see in this example is it's not that you just catch the same neurons with 200 microamps, and then you just add more neurons to it. You actually get a different ensemble of neurons, but there is some overlap. And the question was, is this overlap completely random here, or is there actually some sort of mechanism behind it? So we did a comparison to chance, and essentially this just shows you that for all the brain regions, only the therapeutically effective intensities, they are actually having an overlap that is not random. So before, like with like a current intensity like 100, that you don't really see a response to, like the activation and the overlap is randomly. So as this was a lot, I know, like, so as a summary, I hope I convinced you that these SubUp3 mice are a good model, and that we can actually also use them to study DBS effects in an OCD mouse model. And what we definitely have seen like multiple times is that these effects on brain and behavior of this internal capsule stimulation, that they scale very nicely with charge. So both in the brain and in the behavioral level, which is not true though for frequency, which is interesting because that is also what is done in the clinic. So we actually kind of validate the DBS approach, like in the psychiatry department, like don't change the frequency, like, or scale it up because it's not going to help you. What definitely is somewhat surprising is how broad these effects are. Even though we are stimulating a big fiber bundle, we thought we'd get something that's a bit more specific to specific regions. So we're actually dealing with something that really kind of modulates the entire brain, it seems, or at least like all the fibers that are going through the internal capsule. But not everything has changed. So we have like this, this scattered but overlapping sets of neurons that we talk to with DBS, but we don't touch at all like how they talk to each other, sort of on a local level, so to say. And importantly, like what they do in terms of response, being inhibited or excited, is constant and stable as well. And also so is the number of neurons that are excited and inhibited in the same brain region. So we think that this sort of like, enables an anti-compulsory effect without sort of really changing the entire brain, because you're applying a very massive stimulation, but you don't get any impairment in the patients. It's just actually like a fairly specific effect. And this might be how it's achieved. And this is still somewhat preliminary, we're still adding N to this. It seems like some regions are sticking out, and it seems like the dorsal striatum is special in the sense that it doesn't really have any inhibited cells, and the OFC is strongly activated, which may have, really sort of like a preferred sort of effect, actually altering OFC information processing with the striatum, and the rest of the brain maybe just being modulated or tickled a little bit. And with that, I'd like to thank you for your attention, and especially Bastien, who has done like this huge amount of work for this project. It was very impressive. Aishu Pathasarati has helped with the analysis. We're doing this together with Damian Denise, and we're also looking at human patients. And Tycho Hochland has helped with these microscopes, and Annie van Beest has helped with the wide field imaging. So thank you very much.
Thank you very much, Ingo, for a very nice presentation. If you stop sharing, then the audience can see you again. We have a few questions. Odile, do you want to start off with the first one?
Yeah, maybe I can start with my own question first.
Also possible, yeah.
So thanks a lot, Ingo. It's a brilliant presentation. I really love it. I really liked your thought about us being a neuron and being excited or inhibited in response to stimulation. So that's a funny analog, I think, to people working in science. But I was wondering, this rigid response to stimulation, is that only on the short term or also on the long term? Because these patients get stimulation for years. And I think you presented the direct effects after short-term stimulation. So what do you know about the long-term effects?
Yeah, that's a very good question. And we've thought about this a lot. Because yeah, like what I've showed you now is really just like a stimulation that goes on for 1 minute. And we see stability in that 1 minute. But at this point, we do not know what happens, let's say, if we would stimulate for 1 week. So this would be one of the next steps to go for, to kind of see, like, can we implant this like for longer periods, and can we look at the behavior and the imaging for much longer periods that are more akin to what a patient goes through. So we've landed a first stab here at the response of the brain, but that's definitely something we have to do. It's a bit more complicated because comparing it to some sort of control or baseline is harder, because you're just having the simulation switched on for so long. So you really have to, like, and we're trying to do this now, to look more at like dynamics of the neurons, like just like over time, and comparing that with behavior. But yeah, it's a very crucial thing that we definitely have to do.
Yeah, thank you. So then I will mention one of the first questions that's in the list.
Yeah, go ahead. The top two.
Arba. She is asking, in my eyes, varying DBS settings seem to have immediate effects on grooming. So are the effects of varying DBS settings as immediate as compulsive behavior in human patients? It's a bit in the same line of question.
Yeah, that's also like something, yeah, like that's extremely crucial, of course. So in patients, like it's a bit difficult to answer. Like the simple answer would be, in patients, it takes longer for the anti-compulsive effects to take place. So actually in the psychiatry department, they're now thinking that like the immediate effect is more on the emotional level, anxiety, confidence being changed, and that then kind of trickles down to anti-compulsory effects. But like what also happens in patients is that if you actually found the first, like for the first time, the effective parameters you want to use. So often you have to like go for months, maybe up to a year even, to find the right parameters. So it's kind of hard to determine if the effects are anti-compulsive immediately, in a sense, because you also don't have the right parameters right away. And what you see is, I think that if you, there's a few people that worked with like patients and switch them on and off, that when you switch them back on again, the effects are much more immediate. So it's tough to answer this question. What we should do in our work is, I think, also to check for the anxiety or anxiety-like behavior of the animals to see whether that might even be like preceding the intercompulsive effects, and maybe that's something that is like that the compulsive intercompulsive effects are dependent on, which like what is assumed for the patients now.
We have another question from Jan Boy. Clinically, DBS is used in treatment-resistant OCD. I assume that your model that's used, the CPAP3 model, is a model of OCD per se. Is there also an animal model of treatment-resistant OCD?
That's a good point. Yeah. So I mean, of course, like I mean, if the first part of the answer should definitely be like these mice don't have OCD. Like they definitely experience a number of behaviors that like are, you know, similar to what OCD patients have, and we also see some brain changes and they respond to the same medication and so on. But like all in all, like I think of them more as animals that are, you know, they're showing compulsive behavior, and we are sort of forcing them to be compulsive by a gene deletion that messes with like the same brain regions that are affected in OCD. So in that sense, I would think, yeah, that like we're probably more on the end of refractory OCD patients with these mice in general, because like they also have this gene deletion from the get-go in their development. So it's like, it's not, you know, that they are slowly developing these effects. Like, so, but yeah, it's something we could get at this potentially. So like, interestingly enough, we do see variation in how much these animals groom. So it's not just a gene deletion, there's also some interaction with their environment. So we could try to, you know, and we've done this in the past for behavior, to split them into animals that are not grooming as much and animals that are really grooming to the point that they lose their fur, because that is like really obviously like very negative consequences for them. So we could see whether maybe like the DBS also like acts differently on animals that are like more affected than others. So but for that, we'd have to really increase our cohort, but it's a very interesting point.
We have time for one very quick question and one very quick answer.
And then maybe best Wim's question, because the other one is more maybe for Martijn.
Yeah.
So Wim van den Brink is asking, do you think we could use the CPAP3 model for the study of the effects of psychedelics in OCD?
Yes. Simple answers. Yeah, I mean, like, yeah, I mean, like, again, like, I don't think they have OCD, but we could definitely see, you know, like how their condition, which is very much alike OCD, I think, how that reacts to psychedelics. I mean, and it hasn't really, I think people that conducting these type of basic research based with animal models, there's not really much going on yet. Like, I think like the research in humans when it comes to psychedelics is like ahead in this. But it would be extremely interesting, I think, to see, because we can, especially if we can look at these ensembles of individual neurons, to see how their population activity in general is changed by psychedelics, which I would assume is something, again, like I don't think that we will see something that like entire brain regions are deactivated, but we'll see like how these neurons are talking to each other, like how that's affected. And that's something that could be done specifically in an animal model and not in humans. So I think that could be quite interesting.
All right, thank you very much, Ingo. It was really, really interesting, but stick around for the discussion at the end. Odile, can you take the next part? Yeah.
So then we move on to the second part of the webinar. I would like to introduce to you Martijn Figee. We once started as very junior clinicians in the AMC long time ago, and he's trained both as a clinician as well as a researcher in the AMC and the supervision of Demi and Denise. He got awarded the Ramar Prize of the Dutch Association of Psychiatry after finishing his PhD, and then took the step in 2018 to move to New Amsterdam to see what's better there than in Amsterdam. And it's really great that, being a former CIA partner until recently, to see your work back here in the webinar. So looking forward to your results, Martijn.
Thanks. Oh, yeah, there it is. Okay, sorry for that technical interruption. So pleasure to be back in the Netherlands, although unfortunately not in person, but this worked great as well. Ingo very beautifully explained the sort of what of deep brain stimulation, which is a little harder to do in humans. But in humans, we can actually show, which I will do in the next 20 minutes, where to stimulate. And I'd like to argue that we may actually be able to work towards a more personalized way of stimulation if we really know where to stimulate for which patient with his or her symptom constellation. So by now you all know what deep brain stimulation is. It's of course, so this is how it looks in human. You implant a neurostimulator in the chest, which sends constantly, because it's in night and day, year after year, little pulses to the brain in a specific region through a lead with 4 contact points, usually on both sides of the brain. And these are the targets in the brain that are most often used for obsessive-compulsive disorder. Subthalamic nucleus, which is also used for Parkinson's, but for OCD we use the more anteromedial part of the subthalamic nucleus, the median forebrain bundle, which is a little deeper, mostly used for depression, but also for obsessive-compulsive disorder, which is close to the brainstem. And then the red target, which I will focus strictly on today, the internal capsule. There's different names around. I just want to briefly mention, I'm going to stick with ALIC, anterior limb of the internal capsule, or internal capsule. But there are also names around like ventral capsule, ventral striatum, because part of the lead sometimes touches the ventral striatum or the nucleus accumbens, as I will also show you in a minute. And there's also people using the bed nucleus of the stria terminalis, which is a little more posterior and deeper, but it's definitely also includes ALIC connections. And in fact, even the subthalamic nucleus or the median forebrain bundle, as you will see throughout the talk, are having connections with the ALIC. So in the end, these 3 effective targets for OCD are actually connected through a network. And then the question is, of course, where to put your lead if they all impinge on the same network. If you look at the ALIC numbers, or the combined VCVS ALIC numbers worldwide, there's actually a good response, as Ingo already mentioned. It's like 60% of these highly treatment refractory ill patients actually do have a meaningful response, which is great. And there's also a nice separation between real stimulation and sham stimulation. There's a couple of randomized controlled trials, including our own study in Amsterdam. And you can see a difference between 25 or 40% in the white box decrease. This is the largest study I think done so far here in our own institute in Amsterdam by Damien Denise, and this was mostly analyzed by Ilse and Roel, who may be on the call as well. And you can see again sort of a 50% response rate, which is great, right? But it could be better, which is something we're trying now, of course. And also notice that there's a huge drop in the Hamilton anxiety and the Hamilton depression. So 50% is great, but how can we do better? One of the first thing, again, I'm going to drag my, yeah, there it is. One of the first thing to ask is, like, do we need to be at the ventral striatum, like the gray matter, similar to, for example, what we do for movement disorder, where usually gray matter nuclei are targeted, or is it more the ALIC, like the internal capsule connected to the ventral striatum? So this is a study done by Pepijn van den Munkhoff, the neurosurgeon in Amsterdam. And he actually showed that it is actually the ALIC that needs to be stimulated, as Ingo also mentioned. So you can see that traditionally the lead was implanted with the lowest contact in the nucleus accumbens. But if you actually plot the Y-box scores or the Y-box improvements onto where the actual activated contact is, it is actually always the ALIC and never the accumbens, so, or the ventral striatum. Interestingly, some leads ended up being in the GPE, like the external pallidum, with also some response. So there is some indication, and this will be important later on in the talk, that it's definitely the ALIC, but it may also be sort of a co-stimulation of the GPE. So now that we know it's white matter, where in the white matter, which white matter bundle should we target? Which is a different ballgame, right? We thought we should angle or target the lead specifically into a gray matter nuclei, which is fairly easy for a surgeon. But going after specific white matter bundles is a little harder, because as you can see here, these are 12 patients. This is a study done by Luca Librand, also here in Amsterdam, where you can actually see this is probabilistic tractography in 12 patients that have been implanted with DBS. The leads are in yellow, and you can actually take away from this picture that every patient has a very individual course of their white matter bundle. So he focused his analysis on two bundles. One bundle going through the ALIC, which runs a little bit more laterally, starting at the ventral tegmental area in the brainstem, and then going to the prefrontal cortex, and that's in blue. And then there's the red one that starts in the medial anterior thalamus, and then going to the prefrontal cortex. And you can see that each patient has a different course of their bundles, which again is challenging for a surgeon to target that. What you can see in this analysis is that the lateral track was actually, because the better responders are up and the lesser responders are down, are below in the slide. So you can see the more close the lead was to the blue, sort of the lateral bundles coming from the ventral tegmental area, the better the response was. There was also at the same time a German study where they actually found the opposite, where the more medial thalamic bundle was actually more effective. And this is interesting going forward, because you may actually, for different patients, choose a different strategy. I was very much inspired when I came to New York at the same time that Helen Mayberg and Ki-Sung Choi from Emory came to New York, by their work, where in depression, targeting the subcallosal singlet, again, from this idea that you needed to target a gray matter area, they found over time that it was actually not the gray matter of the subcallosal singlet, but it was actually a combination of 4 different white matter bundles that gave them their best response. And when they started to use that information to prospectively target, and now even showing up these bundles during surgery to sort of confirm that we're in the right place, the response rates went up from 40 to 73 to now even 80%, which is really amazing. In fact, I think what I've seen so far is that deep brain stimulation for depression works pretty well. However, you all know that it's still experimental. It's not approved yet, and I think that's awaiting a more convincing study. But I can assure that will definitely come, especially if we use this more targeted approach. But inspired by this, I started to look in New York, like at the optimal white matter network for OCD. The first thing we did, because we just had a couple of patients in Mount Sinai with OCD, is look at other groups. This is work from Andy Horn here in Germany, although he now works in Harvard in the US. And he took different groups in Europe together, different patients, ending up with 33 patients. And he used a normative tractography, like group tracks, and then he overlay the patient's individual VTAs, the volumes of tissue activated, to see how they correlated with these normative tracks. And you can see in the top picture, those are all the stimulated fibers in all patients, regardless of whether they respond or not. And you can see that ALIC stimulation engages like a wide area of prefrontal cortical regions and also various subcortical regions. But if you look at the bundles that were only correlated to response, to OCD response, you'll end up with with a fairly smaller amount of more focused prefrontal cortical regions. And then you can see the same regions subcortically that we also showed in our 12 prospective patients in Amsterdam from the brainstem and the thalamus. So these are the first 10 patients that we implanted in Mount Sinai. You can see a 70% response rate, which is a little higher than is generally seen, but this is a small group, right? So that can be a statistical thing. This was work analyzed by our psychiatry resident, Andrew Smith. But it's interesting to see that they responded so well. We tried to see if we could replicate this meta-analytic bundle of this multi-European cohort, and indeed we did. So here you can see the leads of all these patients, and in red which contact we activated. You can see that the leads are differently angled. Some are still angled towards the accumbens, others are really more angled at this bundle or at these two bundles, and you can see that the active contacts are indeed in the red bundle that was in this other group associated to a therapeutic response. You can also see that the bundle courses from the brainstem, passing by the subthalamic nucleus and then to the prefrontal cortex. There's also a blue bundle that was actually negatively correlated to response, which seems to go to the temporal cortex, maybe to the amygdala, and doesn't give you a good response. Here you see a correlation graph, and you can also see that, like, one of the poor responders that isn't on the graph, so it doesn't have much engagement of this track. And you can see that actually here, this was a rabbi who was compulsively praying. And interestingly enough, he did have some anxiolytic effect of the deep brain stimulation. And you can see indeed that where we stimulate does engage some of his probably amygdalofugal bundles that we've seen before were potentially negatively correlated with response. But indeed, in this patient, it didn't improve his compulsive praying. And you can also appreciate that the brainstem and thalamic bundles are very thin. So when we modeled co-activating one contact up, you can see now this bundle is nicely engaged. And in fact, that immediately resulted in his compulsive praying going down to under a minute. We had him say a prayer before and after, and it was a difference between like 15 minutes and less than a minute for one specific paragraph. And so you can see that if you use this very targeted approach, similar to what you can sometimes see with Parkinson's, where tremor goes down immediately, you can actually elicit the same acute responses in psychiatric patients if you really know where you're stimulating. This is a good responder who's right on this track, as you can see here. And this was actually one of the patients that we started prospectively targeting. So now our neurosurgeon, Brian Coppell, uses Kisung choice imaging, like probabilistic tractography, 7 Tesla imaging, to target these leads, which is, again, challenging. It was also surprising for a surgeon that when he started doing this, like on the lower row, you see the sort of the traditional targeting where you use a point, an anatomical landmark. But now when targeting on these white matter bundles, you actually end up in very different coordinates in different patients, which again, for surgeons is challenging, but I think it's definitely the way to go for psychiatric patients. Again, unlike what we're used to do in neurological disorders, where we anchor at gray matter structures. So where in the cortex do these ALIC tracks connect to? Because that might give us a better clue of where to go, or maybe even where to go for individual patients. And here we used to work from Susan Haber, who's done very nice studies comparing tracer methods in monkeys with human tractography, where they sort of segregated the ALIC into bundles that are going to the ventral medial and the orbital frontal cortex. Like the most ventral parts of the iliac. And then the lateral parts are going to VLPFC and DLPFC, whereas the medial parts are going to DMPFC and dorsal ACC. And this is actually something that we were able to replicate in our patients. So when we used all the VTAs, the volumes of tissue activated in all of these patients with DBS, indeed, we observed that they were actually engaging these same regions that Suzanne Haber found in her anatomical studies.
Martijn, sorry to interrupt. I think you have to speed up a little bit. There's 5 minutes.
Okay. Do we need all those fibers to get a good response? Well, if you look at responders, it seems like they all engage these different bundles. However, if you compare them with non-responders, you can see that the dorsal ACC, the DLPFC, and the VLPFC are more engaged in responders than in non-responders, whereas these medial or ventral cortical bundles are actually similarly engaged, both in responders and non-responders, suggesting that you may not necessarily need them. And this is also shown here, where actually the VLPFC was positively correlated with change in Y-box, but the VMPFC was negatively correlated. So now that we know which cortical regions are important, DLPFC, VLPFC, potentially also dorsal ACC, where subcortically should we be? Because now you can still, you have a bundle, but you can still place your lead at different places of this track, which is actually done, right? Like the anteromedial STN, which is also connected to these to these cortical regions, or the iliac, or maybe even the ventral striatum. So we did another analysis. This is Josue, our neurosurgical fellow, specifically focusing on subcortical tracks, which is a little more challenging to envision with tractography, but he was able to do it. And there we saw that on top of these cortical regions, we actually have to engage the thalamus and the pallidum. And we also saw some non-response in the ventral striatum and the amygdala. And if you distract these two, like the responders from the non-responders, this was done in 11 patients, you end up with a critical bundle from the thalamus and the pallidum, so this pallidothalamic tract, to these cortical regions, and another bundle from the subthalamic nucleus. And that's interesting because those are exactly part of these indirect and hyperdirect basal ganglia prefrontal pathways. The indirect bundle being involved in inhibitory control, which is definitely something that we know is involved in OCD, and the hyperdirect pathway from the STN involved in switching and moving towards a different behavior or different thought, if you will. So I think for a very targeted approach, we may need to be at the confluence of these two bundles. And then we can even be more selective and try with directional leads to steer the stimulation towards these specific bundles and see if we can use that for an individualized approach, which is actually something that seems to work. If we model this again onto tractography, you can see that with ventral stimulation, you can engage these ventral regions, and then with more lateral stimulation, you can specifically engage dorsal lateral and ventral lateral cortical regions. And with medial stimulation, the medial segments of the lead, you can actually go for these medial bundles. And this is something we did in this patient. This is a patient that has difficulties walking through doors, and he has to compulsively walk back and forth, sometimes for hours. And you can see if we very selectively stimulate his VLPFC bundles, this was right after surgery, you can see that now he's able to walk through doors. He's now 1 year later, he's doing very well, but we also ended up co-stimulating his dorsal ACC bundles. But this is just to show as a proof of principle that if you want, you can actually very selectively influence specific aspects of the OCD. This is a patient where intraoperatively, you can see right after we stimulate his ventral contact, he starts to smile, he starts to notice being more motivated and energetic, but no effect yet on his OCD. And again, if we specifically engage his ventral lateral contact, you can see that he also starts reporting having less compulsive breathing, which is shown here again. This is the patient again a few weeks later after surgery. Again, ventral stimulation.
I felt kind of like in my heart, like I don't know.
Oh yeah, like a little.
Yeah. I'm like laughing now.
So he becomes very giggly, a little unnaturally. It feels like he's on drugs. And when we specifically stimulate again his lateral bundles.
I'm not even compulsively breathing. It's almost gone. Yeah, definitely less. So like, wow. But that was a good setting. The compulsion is virtually gone. Like I'm not compulsively breathing, and
right. So we get a very selective effect on this compulsive breathing without this extreme elevated mood effect, which may be relevant for example, mood disorders. But for this particular patient, it seems to be less beneficial. You don't need it because you actually have the opportunity to selectively engage his specific compulsivity. So this is my final slide. We're now working towards actually testing selective stimulation, but when we're using different tasks as sort of our circuit-specific outcomes, like a gambling task or an inhibitory control task. And then while playing these tasks, we're actually running through these different circuit selective settings to see if we can actually elicit specific outcomes. And so far, we've already seen some promising results where, for example, medial dorsal ACC stimulation gave us a very specific behavioral outcome that was different from the other contexts. So this is my conclusion. I think DBS is a highly effective treatment, but it can actually be more effective, most likely, if you very selectively engage specific circuits. And we may actually be able to work to an individualized approach. So thank you. This is my lab, or actually all the doctors I work with and the scientists, and I'd be more than happy to take some questions.
Thank you very much, Martijn. Very interesting. If you stop sharing, yeah, great. So we'll start with some questions, but please go ahead and enter your questions in the Q&A and we'll pick them up as we go. Odile, you want to kick off?
Yes, so maybe the question forwarded by Mona Hove already is a good one to start with. It was already questioned in the previous lecture, but I think Martijn can respond to that. The question is, I believe that some of the newer DBS electrodes implanted in the OCD patients are also able to record activity at the single human neurons. Do you know if we see the same sorts of effect there in terms of excitatory inhibitory balance?
It's very early on, right? Most of the LFP recordings are done in movement disorders. I think so. There are now for the past few years, like experimental IPGs that also allow for LFP recordings in patients. I think actually Tara is a good one to comment on this, because she's done some studies in patients. Maybe Ingo, you want to comment, because we just started recording, but Tara actually has some beautiful results in Amsterdam.
Just to super quick injection. So to my knowledge, like anything we can do with recording devices at this point is recording local field potentials. Mm-hmm. So that is a population signal that consists of the activity of many neurons. So the answer would be that we cannot record single neurons at this point in human patients. What we can do is we can see what the population of them does together. Like that's what the LFPs are. So like they are a great promise, but we're not going to get to, at least for now, to the single neuron level.
I also had a question. It is, of course, entirely not my field, but what I was wondering is, you show really great promise with all these techniques, but what happens if a patient undergoes all this and there's no response? What do you do then, counseling-wise, or?
I mean, as I showed, fortunately, it is a minority. but definitely we do sometimes have responders. I mean, it's interesting, in the Amsterdam group, we have often come across patients that don't have a perfect response according to the Y-books, but still are happy with the result because they have some sort of mood change or anxiolytic effect. Like I showed you with the rabbi who didn't see any change in his compulsive praying, but at least he was less anxious, right? So again, there's probably different aspects of OCD that you can influence with deep brain stimulation. Of course, you want to ideally influence all of those aspects, but it's very rarely that a patient doesn't have any benefit. But if they do, you can choose to explant. But it's unfortunately very hard by then to treat them with all, because these patients have already exhausted the whole treatment protocol, including all kinds of medications and cognitive behavioral therapy and TMS. So it's hard to then find something else. DBS is actually the last resort.
Then a question from Martijn van der Boom. Do you think that conceptually the anti-compulsivity effects can be attributed to a single fiber bundle? Do you think multiple are involved?
I think compulsivity is a complex construct, right? Which involves things like positive and negative reward prediction, inhibitory control, reversal. So all these things we're testing now with these tasks. So there's different aspects of compulsivity, and maybe each of these aspects, and this is something I believe, are actually coupled to specific bundles, because of course we know things like reversal learning or inhibitory control can actually be attributed to specific circuits, but not compulsivity as a construct because it's too broad.
There's a question from Annick Broekhuis, which I think is also a good one to clarify. The question is, you mentioned that there are ways to visualize white matter tracks during surgery. Could you elaborate on how that works? So I think, what is the order of events? What do you do when?
So you need, of course, first an image of where the lead is, which you take intraoperatively, because you have CT scans during the surgery. In fact, the surgery is sort of applied in a CT scan, which is called the O-arm. And so with that scan, the imager can actually overlay the tractography of the patient onto that lead. It takes like 15 minutes to process. I mean, if you want to like a high density image, it takes like days, but you can have sort of a quick shot of your tractography overlaid on where exactly the lead is, and then the surgeon waits until the image is in, and then we sort of check if everything fits, and if not, we can still make adjustments and then check again. It's still a little bit of a lengthy process because again, there's always sort of a 15 minutes delay, but that's as good as we can get it now.
And the MRI scan itself is done before surgery, right? I think.
Yeah. So the MRI scan is always done before. Yeah. You can't do it afterwards anymore, even though we're all working towards that. So these newest implants are actually also MR compatible. So people are now working towards also getting post-surgical MRIs.
So a question from maybe best first to do the question by Frederik Hartereiter. It's already from the first lecture. If I'm correct, in Parkinson's disease, we already adjust the stimulation according to the symptoms of the patients. Isn't there a safety margin within which you can clinically test on OCD patients?
I was also curious what Ingo's answer would be there, because I mean, clinically, we do see that if we stimulate, more is not always better, right? So there is a certain threshold which aligns with what Ingo showed, where patients start to respond. But we also see sometimes if we respond, or if we stimulate with a too high amplitude, patients develop side effects, including things like impulsivity or anxiety or feeling restless. So I was wondering if Ingo has actually seen on a neuronal level if there's such a thing as too much.
I think it's a good point to actually kick off the discussion together. So Ingo, please join us, and you can stay with us.
Yeah, that's a good question. We tend to focus on like as Martijn just said, like the threshold of like one is actually like one is getting to the point of the animals are responding behaviorally to the stimulation. We have at some point in the very beginning like tried out to go a little higher, and I'm not sure if that would be the same response in humans, but like in animals at some point, if you crank up the current, let's say to another, you know, like maybe double it even or something, like you will get seizures. Like the animal will have seizures. And from what I remember, this is just like not very systematic study, but I think we also saw brain damage at that point, like at the electrode tips. So if we now take out the brain and check, we can see the electrode track, but it seems like the neurons near the electrode are fine. But I think if you crank it up more, you will get A, seizures, B, neuronal death. But we haven't really explored that, and fortunately we didn't have to. It seemed like the amounts that we applied actually were sufficient to really bring down like the compulsive behavior sufficiently.
There's also a question that I think would fit both of you. So I'll read it out, but it would be great if both of you could give your views. You showed internal capsule DBS might be stimulating multiple striatal pathways at once. What do you think about the working mechanism of DBS in these pathways? Is it affecting the fiber bundles themselves uniformly, or instead is it affecting the gray matter targets of these fiber bundles differentially?
So maybe I'll start like I mean, like what is to me is fascinating is that, I mean, also what Martijn was saying earlier about like the work in depressed patients, that it seems like the field is moving towards white matter targets also in other disorders, which automatically means that, I mean, in a way, like you're affecting something that's gonna, you know, like your local stimulation is gonna affect regions that are much bigger than just like the immediate effect of the electrode like current that is actually like applied to the tissue. So somehow it seems like with this, and maybe it's truly a neuromodulatory technique because of that, that you actually like you're affecting these fibers that then affect a bunch of different places in the brain. So you're not really affecting specific regions. So I think, I mean, my answer kind of like, I mean, if I had to guess, I'm thinking from what we've seen so far in humans and in animals is that you know, it has to be like a good chunk of the brain that you're affecting and not just like a specific area. I think that answer will be maybe coming. There are some groups in the world that are trying to be more specific. They're going the other way. So we'll see what they're doing because they want to target the cortex specifically. But to me, what seems to be happening, at least in like what we've seen so far, is, I mean, you're applying a high frequency stimulation, 130 hertz, 120 hertz. Almost no neuron in the brain can fire at that frequency. So you're actually not entraining neurons to do the same thing as your stimulation. You're somehow applying some sort of like, you know, like modulatory tickling event. And that seems to be specifically like good at fibers. Like, so like I think what we have to do is to really check what does an electrode with that high frequency stimulation, what does it do to a fiber? Like we don't know the answer to that actually. Like, so which is something that might help us then, you know, in translating this back, if we actually know what we should be seeing in a fiber. But yeah, but to go back to that question, I mean, it seems like to me, it's that whatever fibers you're closest to, you affect them uniformly. So it doesn't seem like from what we've seen so far, that there are specific regions that stick out a lot. Like Martijn, what do you think? Yeah, no, I mean, we've modeled stimulation towards specific bundles, as I showed. But these are models, right? We can't specifically test, or we can, and in fact, we're trying to do now. But this is still mostly LFP and EEG, because you can't, for example, test it with functional imaging because the leads are now in. But we've modeled, and these are neuronal models that are assumed to be pretty accurate. That if you use specific amplitudes towards specific segments of the lead, lateral versus medial or dorsal versus ventral, you can actually
engage specific bundles to specific cortical regions connected to specific subcortical regions. So that's a model. I don't know if that's going to be therapeutically necessary. As I showed you in these patient examples, you can actually use it to influence specific aspects of compulsivity, but it doesn't mean that that's necessarily what the patient is responding to in the end. In fact, in these two examples, we ended up stimulating different bundles. But I think it's just an interesting model, and it's even a trend diagnostic model where for different disorders involving compulsivity, or at least involving these circuits, you actually have the choice to selectively stimulate. That's what I believe. It's just going to be quite some work to really get there.
So the answer is kind of in the middle, maybe, right? Like that we are uniformly affecting fibers, but only specific ones. So that means multiple brain regions, but not everything.
Yeah.
So maybe there's room for one very short question by me. So we are in the field of clinical neuroscience now really trying to get more personalized targets, and all these techniques really contribute to this. But at the same time, we also would like to keep care financially good enough so that we can treat enough patients. And I was recently reading the meta-analysis where they compared the very old surgery with the deep brain stimulation, and that the results on the treatment outcome are not that different. And since we now have these techniques like diffusion imaging, can we maybe go back to surgery when we know more precisely where to cut, instead of using these very expensive techniques in say, stimulating the brain for 20 years with very expensive materials? So do we go back to surgery?
Yeah, the more targeted we are, the more precise we are becoming, the less we need to toggle around with these different stimulation settings, right? And we can even make a specific lesion there. We're not there at all now. So now I would definitely favor deep brain stimulation, because you still have a wide array of possibilities, as Ingo showed, where you can actually nicely engage specific aspects of the circuit. Whereas with a lesion, it's one cut, and you better make sure that you're there. And you're right, the efficacy is similar to DBS as it is now, but I think we can definitely bring DBS from this sort of 50% to like 80% or 90%, as some studies in depression have already shown.
Yeah, but that might also be the case with more precise surgery. So
yeah.
And maybe the individualized approach will actually then kind of in the future lead to the point of like, oh, this patient should get DBS, but this one may be a capsulotomy or a specific lesion. Like that may also be an advantage.
Yeah.
Okay, thank you very much, everyone. I have to cut you off, unfortunately. I will just share my screen for a final point. So thanks, everyone. Thank you to Martijn and Ingo, of course, for coming here, especially Martijn so early in the morning. And please join us for our next Dn2 seminars. The next one will be on neurovascular disorders on January 21st. The setup will be the same with a host and a co-host and 2 speakers. And keep an eye on our website, our Twitter, and our LinkedIn. So thank you very much and have a great day.